Hi, I'm Dr. Leslie Blankenship-Williams, and in this lecture we are going to start talking about the process of gene expression. Now, on the most basic level, gene expression is essentially the process by which the genetic information in the cell, that would be DNA, somehow gets expressed into whatever that DNA codes for, which in most cases is a protein. So, in a simplistic way, gene expression is basically starting with DNA and somehow getting to whatever that DNA codes for. As an analogy, let's think about your computer. So you have a computer and it has software installed. And at the most basic level, the software is written in binary code, a specific sequence of zeros and ones, like so. But you don't really ever see those zeros and ones. When you double click an icon on your computer, let's say for a word processor or an internet browser, zeros and ones don't pop up on your screen, but rather the zeros and ones get expressed into a useful program. So what's really happening? The zeros and ones are getting translated into a word processor that you can use, or a photo editor that you can use, or an internet browser that you can start playing around with. So if we were to think about that computer as an analogy to gene expression in a cell, then these zeros and ones are going to be the code, like DNA, and what you see on the screen would be like the protein, what the zeros and ones code for. Okay, so we'll be getting into the process of gene expression in a little bit more detail later on. But now I want to turn and talk a little bit more about DNA and how it exists in different types of cells. So in your workbook, you were required to learn about the structure of DNA. And so you know that DNA is comprised of a sequence of nucleotides, and that each nucleotide has a sugar, a phosphate group, and something called a nitrogenous base. And there are 4 different nitrogenous bases. For DNA, we have A, C, G, and T. Now, DNA exists as a strand of these nucleotides. But in the real world, DNA is never just one strand. It's always a pair of strands joined together by a series of hydrogen bonds. So one way that we can actually depict this is to think of this line as what's called the sugar phosphate backbone, and these steps as the nitrogenous bases. So let's say that I have A, C, G, G, T, A, T. I'm just making stuff up here, specific sequence. Okay, in reality, that DNA does not exist as a single strand like that. There is always a complementary strand. In other words, the other half. And we know from base pairing rules that A always pairs with T and C always pairs with G. And the cutesy way to remember that is the curves always go together. So C and G both have curves in them, they go together. A and T has straight lines, and so they go together. So using these base pairing rules, we can actually predict what would be on what is called the complementary strand, the other half. So up here, I have A. So next to it, I would have T based on this set of rules. C would pair with G, G would pair with C, G would pair with C, T would pair with A would pair with T, T would pair with A, like so. Okay, so in reality, DNA exists in this double-stranded arrangement, and hydrogen bonds between the bases end up holding the strand together actually pretty tightly, like so. Now it turns out that that double strand is not straight, but rather kind of wraps around itself and spirals. And this is more commonly known as the double helix. And so you can see a double helix here, and you can see a double helix here. Now note that this is consistent regardless of whether or not you are a prokaryotic or a eukaryotic cell. So DNA is always double-stranded and always exists in this double helix formation in both prokaryotic and eukaryotic cells, basically any living cell. Okay, so now that we've covered the way that they're the same, let's talk about some differences. A long interrupted strand of DNA is called a chromosome. And when I say a single strand, I want you to know that I mean a double helix. So it's really two strands joined together. So that's going to be our chromosome. In a prokaryotic cell, the chromosome is going to be one circle. So you've already seen me draw this a few times. But there is one circular chromosome. Okay. In eukaryotic cells, the chromosomes are linear. And typically, though not always, but typically, they are found in multiples of 2. In other words, an even number of them exists in any cell. And not just anywhere in the cell, but specifically in the nucleus. So here, the chromosomes are linear, and they are usually found in multiples of 2. So here I have 4 chromosomes. Notice I have 2 big ones and 2 small ones. So they tend to come in pairs. In other words, there are 2 copies of each chromosome. What about a human? How much does a human have? A typical human cell would have 46 chromosomes, and there are 23 types. And we are so clever in how we name them. We call them chromosome 1 and chromosome 2 and chromosome 3 and chromosome 4, etc., 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 all the way down to chromosome 22. And then instead of saying chromosome 23 for the last set, that's going to be your X and potentially X or Y chromosome. Those are called the sex chromosomes. So most people have 2 copies of each chromosome in their nucleus. 1 copy you got from mom and 1 copy you got from dad. So 1 copy comes from the egg, 1 copy comes from the sperm. And so you get 2 copies of each chromosome. So if I take one of your cells, you should have 2 copies of chromosome 1, 2 copies of chromosome 2, etc. The important thing to note here for eukaryotes is that there are 2, and we're just going to put a little n there because it's multiples of 2, linear chromosomes. So n represents number of different types of chromosomes. In humans, we have 23 different types. So we have a total of 2 times 23, or 46 chromosomes. So what I have just described constitutes the majority of DNA that you will find in these cells respectively. For a prokaryote, the single circular chromosome exists in the region called the nucleoid. For a eukaryote, the linear chromosomes exist in the nucleus. But is there more DNA than that? There might be. Let's take a look. It turns out that whereas the vast majority of DNA in a eukaryotic cell does exist in the nucleus, there is also DNA in a couple of the organelles, most specifically the mitochondria, and if you're a photosynthetic organism, also the chloroplasts. So where would DNA be located? Most of it is going to be in the nucleus, but you might also have a little bit in the mitochondria and the chloroplasts. Now I drew squiggly lines, but please note that the mitochondria and chloroplast DNA is not linear. It's actually circular, like prokaryotic DNA. So to summarize, DNA is found both in the nucleus and these are our official chromosomes. But then there's extra pieces of DNA found in the mitochondria, which almost all eukaryotic cells have. And if you're a photosynthetic organism and you have chloroplasts, then there will also be DNA there as well. And the mitochondria and chloroplast DNA actually codes for proteins that are found in the mitochondria and chloroplasts respectively. All right, what about prokaryotic cells? Well, I've already talked about the one circular chromosome found in the nucleoid, but it turns out that there can be extra small circular pieces of DNA that are found outside of that one chromosome. Now, these are optional, and if they exist, they are called plasmids. So you can see the word plasmid right there, and you can see what it's pointing to. Plasmids are optional. I think of them like accessories. So these accessory pieces of DNA are also circular, and they are much, much, much smaller than the single chromosome. Whereas a chromosome might have anywhere between 2,000 and 4,000 genes on it, plasmids usually have less than 10 genes. So we're talking a really small little bit of extra DNA. Why would prokaryotic cells like bacteria have plasmids, or why would they want them? Well, they give the prokaryotic cell a little bit of extra genetic material that might be useful to that prokaryotic cell. One type of gene that is often carried on plasmids are genes coding for antibiotic resistance. So that's good for the bacteria and bad for us when those bacteria are pathogenic. We will revisit plasmids in future lectures and also in labs 14 and lab 16. So we'll see plasmids again. It's important to note that plasmids are largely optional. Because they are optional, bacteria don't necessarily require them for survival, which means that plasmids can be present and have some sort of advantage, but if they get rid of them, the bacteria don't necessarily die, but then they lose whatever advantage that plasmid gave it, if any. Okay, so now that we've taken a look at where DNA exists in prokaryotic and eukaryotic cells, let's turn to a different topic. Let's talk about genes versus alleles. If you've had a general biology class, then you probably have seen these terms. So a gene, by definition, is going to be a stretch of DNA that codes for some product, typically a protein. An allele is a variant of that gene. Perhaps this is better explored with an example. Many plants produce flowers that have different types of colors to them. Imagine that you are looking at a particular flower and it's purple. Let's go ahead and draw a purple flower here. Now the purple flower exists because there is a purple pigment that is produced and basically expressed in the petals that makes the flower look purple. But let's say that the same species produces the same looking flower but doesn't lay any pigment in it at all. Then the flower would be white. Okay. Both of these are coded for by the same gene. In other words, there is a gene for flower color. But there are two different alleles or variants. there is the variant that actually makes a purple pigment, and then there is a variant that makes no pigment, and therefore the petal looks white instead. So what we have here is two variants of the same gene. So when you're thinking about genes versus alleles, so when you think about the difference between genes versus alleles, remember that all flowers are going to have a gene for flower color, but which color they put. But the different types of colors represent different alleles. So purple or pink or red or maybe even blue or white or yellow, those are all alleles, different variations on the flower color. So that's the difference between gene and allele. Now, why is this important? Well, remember how I said that eukaryotic cells tend to have chromosomes in sets of 2? In other words, 2 copies of each type? What that does is typically give them 2 different alleles for one gene. And we can see that here. We have 2 chromosomes. Those are the long purple stretches that you see right here. And we have one gene, but there are 2 different alleles. So eukaryotic cells, because they have 2 copies of the same chromosome, get 2 shots at a particular gene. Now the alleles that you might find for that gene can be the same. In other words, we could have 2 purples, we could have 2 whites, or they could be different. But my point being that they get 2 shots at that gene. Now why is this important? Let's say that I take a gene for something really, really critical. So I've got a chromosome, and I have a gene, and I'm going to mark it right here, and I end up with a mutation that ends up creating something that doesn't work in one of my chromosomes. In other words, the gene is corrupted and it no longer produces the desired product. Well, as long as the other one still works, you should be okay. In other words, because you get 2 shots at the same gene, if you get a mutation in one of the alleles, the other allele might be able to compensate for that. Now, I'm generalizing here. It's not always the case, but very often it is. Let's contrast this to bacteria. Remember that bacteria have only one circular chromosome. Now, what this translates to is that they tend to have only one copy of each gene, not 2 copies like eukaryotes do. So let's say that this gene that I've highlighted in blue is required for life. In other words, the bacteria need it to survive. And that gene incurs some sort of mutation so that the gene no longer produces the useful product. If there's not another copy of that gene somewhere else in the chromosome, and there might be, but not always, then that bacteria dies. There isn't a second chance, per se, to recover from that mutation. So for that reason, when mutations hit bacterial chromosomes, they can be lethal a lot faster than they might be in eukaryotic chromosomes. Let's put down what we've just learned into a table that compares and contrasts the DNA arrangement in eukaryotes and prokaryotes. So both eukaryotes and prokaryotes have double-stranded DNA. And this DNA represents their genetics. So they use DNA for their genetic code. Eukaryotes have multiple linear chromosomes. And again, usually these are in multiples of 2. Prokaryotes have one circular chromosome, Eukaryotes tend to have 2 copies of every gene. Prokaryotes tend to have 1 copy of each gene. Eukaryotes do have extra-chromosomal DNA. That means DNA that exists outside of the nucleus. But their DNA tends to be found in mitochondria and chloroplasts. Whereas prokaryotes have their extra-chromosomal DNA as plasmids. Now we can take a look at what we do with DNA. So if I have a prokaryotic cell, and there's the DNA, one thing that I can do is I can replicate that DNA. And we would see this if you were going to divide by binary fission or if you were going to go through endosporulation. Remember, most bacteria are not capable of forming endospores, but if you are, then that's a reason why you would replicate your DNA. Now, there are a number of enzymes that are involved in DNA replication, but the big one is called DNA polymerase. So DNA polymerase is involved in replicating the DNA, either through binary fission or to get ready for sporulation. Now, that's one thing you can do with your DNA, but just copying your DNA over and over again without using it is kind of pointless. So let's talk about how we actually use the DNA for its real function, and that's gene expression. So the basic code of DNA is to go from A, C's, G's, and T's somehow into a protein, or whatever the product is. Again, it may not be a protein, it can be RNA, but proteins are most common. So that process is gene expression, and it is actually a 2-step process. So the process of gene expression goes like this. We start with DNA. Now DNA, again, always exists as a double strand. So we can almost draw these railroad tracks here to denote the double-stranded DNA. Then what's going to happen is an enzyme called RNA polymerase is going to read parts of that DNA and make an RNA copy as a byproduct. So RNA is a very similar but not identical nucleic acid. Now the RNA is single-stranded. So unlike DNA, the RNA is single-stranded. Now that RNA basically represents like a photocopy of that gene, a mobile photocopy. Remember, the DNA is kind of locked away on the chromosome. It's not able to move very easily. So the RNA represents a photocopy that you can basically move from place to place. And so the idea is to take that RNA and move it to the organelle known as a ribosome. Now ribosomes, yes, they are organelles, but they are also enzymes. And ribosomes are responsible for reading that RNA and translating it into a chain of specific amino acids. And each ball or pearl is going to be an amino acid. And so we call this a peptide or a polypeptide. And other forces occur where that polypeptide is then going to fold itself up into, secondary structure, tertiary structure, and potentially quaternary structure. And once you have a 3D folded polypeptide, that is a protein. For our purposes, we're really just interested in going from DNA to polypeptide. Now, going from DNA to RNA and RNA to polypeptide are two separate steps, and they have names. So going from DNA to RNA is called transcription. To transcribe something means to copy it. DNA and RNA are of the same alphabet. They are both nucleic acids. So the analogy would be like going from American English to European English. You might throw an extra U in there once in a while into your words, but it's largely the same language. In the same way, DNA and RNA are very similar. There are some slight differences in their alphabets, including a U, as we will see in the next lecture. But transcription means to copy. So going from DNA to RNA is effectively copying the DNA. When you go from RNA to a polypeptide, you're changing alphabets. RNA is a nucleic acid. Polypeptides are amino acids. They're totally different molecules, very different structures, totally different alphabets. So this would be like translating. And in fact, this process is called translation. So again, the way you can remember that translation is the RNA to polypeptide one, is to remember that to translate is to go from one language to another. And here you're going from the language of nucleic acids to the language of amino acids. Totally different languages. All right, here's a quick look at the process of transcription and translation in a eukaryotic cell. Now notice that a eukaryotic cell has its DNA housed in a nucleus. So the RNA is also going to be made in the nucleus. And then the RNA leaves the nucleus and finds a ribosome out in the cytoplasm to be translated. Prokaryotes do not have a nucleus, and therefore do not separate their transcription and translation in the same way. Transcription and translation in prokaryotes happens a lot faster because it all kind of happens at the same time. In other words, RNA is produced and a ribosome grabs onto it as soon as it's coming off the DNA. It's not separated into a separate compartment like it is for eukaryotes. Okay, that concludes this lecture. I'll see you in the next one. Thanks.